So I'm deeply honored and humbled to receive this President's Young Investigator Award from the ASRS Foundation. There's no greater recognition and honor than the recognition of one's peers, so I thank the Foundation for this great distinction. And I dedicate it to the 36 scientists who have traveled this journey with me for the last 15 years, and to my wife, Kameshwari, whose support has been instrumental, and to my children, Meena and Divya, whose smiles have been inspirational to me. Today, I'll share with you some philosophical thoughts on how we can move forward in the fight against AMD by reflecting on the past. This year marks the 20th anniversary of the landmark discovery by David Hinton's group, who showed the presence of VEGF in human CNV. This observation propelled a rigorous investigation of VEGF biology in animal models in the eye, and we moved from lab to label in an astonishingly rapid 8 years, when the first anti-VEGF drug was actually approved for CNV. And more shortly thereafter, even better anti-VEGF drugs arrived on the scene. So why hasn't this triumph been replicated for geographic atrophy? Is the problem too hard? Are we attacking the wrong problem? Or are we using the wrong approach? I don't think the problem is too hard. A mean annual growth rate of 1.8 square millimeters is equivalent to 1 RPE cell dying every hour. So even a modest therapy that saves just 12 cells a day can be monumental. Vas Sadda's group has shown the GA marches towards the fovea at a rate of 0.14 millimeters per year. So saving just 12 cells a day can dramatically delay vision loss by many years and be monumental. Are we attacking the wrong problem? Most therapeutic efforts focus on complement inhibition because of the exuberance surrounding the statistical genetic association of complement loci with AMD. Yet Rob Mullins has shown that complement activation occurs even in children as young as 5 years of age, and that most complement deposition occurs not in the RPE, but rather in the choroid, leading one to question the wisdom of intravitreous delivery of these drugs. In contrast to the rigorous biological studies that led to 4 therapies for CNV, 20 years of gene hunting has produced a laundry list of failed trials for GA. At least 4 complement drugs have failed. And while I certainly hope, for the sake of our patients, that the ongoing phase 3 studies succeed, a Bayesian worldview compels one to be somber about its prospects. If that trial too fails, we as a community have to be intellectually honest and admit that the biological association of complement is not as strong as the statistical association in AMD, and stop beating a dead horse. 2 decades ago, the tyranny of imposing statistical genetics as a litmus test was in full bloom when NIH grant applications from Tony Adamus and others were dismissed as pointless. Today, not only is the biological role of VEGF in AMD firmly established, but we know from Dan Martin and the CAT group that polymorphisms in VEGF and VEGF receptors don't even predict the response to anti-VEGF therapy. Despite these stunning indictments, the altar of statistical genetics continues to demand sacrifice. 15 years after the Human Genome Project, it is clear that medical insights won't just pop out from sequencing genes. Pursuing polymorphisms without considering the sheer complexity of gene interactions, epigenetics, and the microbiome is akin to a geocentric view of human genetics. So are we using the wrong approach? There's a great deal of effort afoot on answering the question of whether GA is a single disease or multiple diseases. And while these classification efforts are indeed laudable, I think a more pressing question is, can a single treatment suffice regardless of whether it's one disease or not? Likewise, tremendous international resources are being brought to bear on cataloging all the genes involved in geographic atrophy. Again, I think the more salient question is, will following the genetics yield a therapy? And I think it bears reflection that in a hypothetical world where everyone smokes, lung cancer too would be genetic. But everyone is aging, so naturally GA has a genetic component. But is that going to help us achieve a therapy? We've all seen pictures like this, and the question is being asked today, an important one: are these different diseases? Maybe, but maybe not. If one were to throw rocks at car windshields, the patterns of the shattering would be tremendously diverse. Does this mean that different things happen to all these windshields? More to the point, Yi Zhang and colleagues at Emory University have performed 2D cellular POTS modeling of random cluster cell death that simulates geographic atrophy seen in humans, suggesting that the underlying problem is a stochastic systems failure, not dissimilar to what we see in other chronic diseases like diabetic retinopathy. We could call all of these different diseases too, and perhaps they are, but yet it's remarkable that all of them respond to an identical treatment. While genomics is powerful, there's still a great deal we don't understand. Here's a spectacular example of our ignorance in this area. The first human to have his diploid genome sequenced was found to be homozygous for Cockayne syndrome, Usher syndrome, and glycogen storage disease. So who was this person? That was James Watson. And the genome of this man says that he should have been blind, deaf, decrepit, and mentally retarded. Fortunately for him and for us, he wasn't. But this vivid example teaches us that we are not our genome alone. Indeed, we need conceptual shifts in understanding how our genes, environment, and culture collude to cause disease. To me, the most important lesson to draw from the translational triumph of VEGF is to search for a final common pathway that is conserved across many diseases. In GA, the inflammasome is emerging as such an integrator of the cell's response to diverse stressors. The inflammasome is now recognized as a key driver of aging diseases including AMD, Alzheimer's, and atherosclerosis. Developing drugs for such targets can be accelerated by embracing the concept of repurposing drugs. To this end, on Friday, I'll discuss our efforts to repurpose a class of anti-HIV drugs for AMD. We're fortunate that AMD progresses very slowly. Indeed, vision often remains good until the very end, resembling the capitulation event seen in financial markets. With such a visual trajectory, and given that GA affects individuals towards the end of their lives, one modest but effective approach would be to simply delay the disease slightly so that it outlasts the patient. Ultimately, I think we can replicate the anti-VEGF success story by replicating the rich bidirectional dialogue in terms of molecular, biochemical, and pharmacological studies that proceeded between the bedside and the bench and that took place the decade before last. Thank you again, and let's conquer GA.